Well, thank you very much, Topen, and thank you very much to the organisers for this opportunity to come to a beautiful city and meet some nice people. So I have to declare some interests. People give my employers money, but none of it comes to me. And I'm also going to mention GRIP, which I co-chair, and the European Network of Paediatric Research, EMA, which I chair. So the starting point is that minimal risk is an important concept because it's one factor which determines which trials we can do. It determines which individual trials we're allowed to do, and it feeds into the laws and regulations about which trials are ever going to be possible. To address this topic, I'd like to give some background and then look at the approaches that have been used and compare their strengths and weaknesses, and then think about a way forward. For me, the way forward is the most important thing. I'm not a lawyer, I'm not an ethicist, I'm a neonatologist. I have to guess which drug to give, I have to guess which dose to give. So for me, the most important thing is to minimize the risks of ignorance. But I appreciate that we have to minimize the risks of the research as well. Just to clarify, there are two types of research for our purposes. First of all, there's research which may involve the prospect of direct benefit to the individual. This might be giving a new medicine at a dose that we know is likely to work. And in that case, we need to do a benefit risk assessment for the individual whilst doing a benefit risk assessment for the group, taking account of the risks for the individual. And when we think about it, most of the research that we do isn't necessarily going to give direct benefit to the individual. It can be argued that phase 1 trials, first in human studies, are not going to be of direct benefit to the individual because we don't know what dose to give. And same in dose-finding studies, that some of those studies are actually not going to be of direct benefit because we may be underdosing or overdosing until we've done the PK studies. The other type of study is when there's no prospect of direct benefit to the individual. And that's what we want to talk about today, and that includes a number of studies that are familiar to us all. It's partly about the design of the study and the procedures that are involved in the research, things like taking samples, taking blood, urine, feces, cerebrospinal fluid, can be interviews, which can be very superficial, or in general pediatric research can be quite intrusive, talking about sexual behavior or drug-taking behavior. Clinical assessment varies in the risk and the impact, and imaging studies vary in their risk and the burden that they provide to the individual. As well as all these procedural and design aspects, we need to remember that giving medicines can involve risk, particularly when we don't know whether the medicine is going to work or what dose to give. So the question we need to ask is, when is it acceptable to do each of these procedures at different levels of risk during a research project? And how can researchers work with ethics committees? And which is a rhetorical question, because researchers need to have very productive research links with ethics committees. And another rhetorical question is, how should definitions of minimal harm be fixed in European law? And as we've heard, this is a live topic at the moment and is the subject of some lobbying behind the scenes. So to bring this together, I'd like to review the concepts used in discussions of minimal risk. I'd like to try and develop a common approach to the issues and then consider some case studies. And the case studies that will come at the end, I'll mention them now so you could be thinking them through as we're going through the material. First of all, a single dose PK study of cough and a cold medicine. When is that minimal risk? When is that not minimal risk? Placebo treatment in a randomized controlled trial, particularly an efficacy trial. And phase 1 studies in dying children. And I'll cite examples from oncology and gene therapy. And do these ever constitute minimal harm, minimal risk? And should they be possible even if they do constitute more than minimal risk? I think it's useful to start with the history because it does explain where some of the positions come from. And as far as I'm aware, the first mention of how risk, burden and harm contribute to discussions of what's permissible come from 1931. The German Ministry of the Interior, who stated experimentation on infants or persons less than 18 years old is forbidden if it will only expose them to a very slight danger. So the first thought was to protect the children and young people at any cost. This thought was echoed 30 years later in the UK by a very distinguished group. Parents and guardians cannot give consent to on behalf of the minors to any procedures which are of no particular benefit to them and which may carry some risk of harm. The College of Physicians in the UK echoed this. Clinical research of children or mentally handicapped adults should be conducted only when the procedures entail negligible risk or discomfort. And this theme was taken up by the National Commission on Research on Children from the States in the late 70s, where they defined minimal risk as the magnitude of harm that is normally encountered in the daily life of healthy children. So this line of thought was crystallized in the Oviedo Convention on the protection of subjects of biological and medical research in 1997. And I'll emphasize the points I want you to read, where it's in this European declaration, where the research does not have benefit for the individual, it may be authorized, and going to the bottom, if it only entails minimal risk and minimal burden for the individual, and any consideration of additional potential benefits of the research shall not be used to justify an increased risk or burden. So this is quite a restrictive definition, and if you match it with the US National Commission's definition of minimal harm, then basically you can't do research on people of no therapeutic benefit if you're going above and beyond the risks that children normally meet as healthy children. And this is an accepted part of the European ethical landscape. Although in an explanatory report, they did expand on what would be minimal risk, and for example, taking a single blood sample would generally present a minimal risk. So that's a little bit more liberal than the United States definition of minimal risk, because healthy children don't have blood samples taken as part of daily life. They also suggest minimizing risk by replacing plain X-ray with ultrasound, which may rule out certain investigations in certain conditions. While it's frequently cited as being an important document in European ethical discussions, it's not been signed by the UK, Belgium, Germany or Ireland, so has absolutely no relevance in those countries. And although it's been signed in other countries, it's not entered into force in Italy, the Netherlands, Poland or Sweden. So many of the most active research countries in Europe have not adopted this convention. And so while it's widely cited, it's not relevant to many of us. There is a different route, and this was hinted at by the same National Commission in the United States, where they introduced the possibility of a minor increase over minimal risk if you're likely to yield generalizable knowledge or it's of vital importance to understanding the condition. So this takes a different position from the Oviedo Convention and says that under some circumstances, if you're not going to do any benefit, you might do a little bit of harm if it's well justified. And this was echoed a couple of years later in the UK by the forerunner of our Royal College of Child Health, in that it may be justified if the anticipated benefits are sufficiently great. So even for non-therapeutic research, many people have argued that there's a need for a benefit risk assessment. This is incorporated in US federal law relating to trials conducted under the FDA's regulations, so that's drug licensing trials, and they've included things like modest change in diet, non-invasive psychological monitoring, obtaining stool samples as minimal risk, which is more restrictive than the Oviedo Convention. And if you're going to go for something above minimal risk, then the harms must be not severe and must be transient. And interestingly, any risk has to be quantifiable if it's minimal or minor increase. So if nobody has ever mentioned, if nobody has ever measured the risk of collecting a stool sample, then by definition it's a major risk. So there are some problems with this sort of interpretation. So far we've found problems that not doing research causes harm to children, and the definitions are inconsistent. There may be a conflict between the Oviedo Convention and US law, and it's not immediately clear that minimal risk means the same thing in all cultures. In fact, it's not immediately clear that minimal risk means the same thing within the United States. This paper from David Wendler's group asked the chairs of institutional review boards how they interpret minimal risk. And this bar chart shows minimal risk in blue, minor increase over minimal risk in red, and more than minor increase in green. So almost all the IRB chairs felt that taking 10 mils of blood from an 11-year-old child was minimal risk. And all the others, bar one, thought it was a minor increase over minimal risk. A magnetic resonance imaging procedure with no sedation was thought to be minimal risk by half, some feeling it was more than minimal risk, and others felt it was more than a minor increase. Repeated blood samples of 10 mils per week for 6 months in an 11-year-old child were felt to be of minimal risk by some, more than a minor increase over minimal risk by many, but not allowable by others. And the initial testing of a drug found to be safe in 500 adults was felt to be more than a minor increase over minimal risk by the majority of IRB chairs, which for me rules out most drug research in children. People have also looked at what are the risks of childhood, and this is the risks of dying from travelling in a motor car, or how many times you get hospitalised during the average car trip, and so on. And there are quite copious statistics about this in order to balance your everyday risk. So this is one approach, which is trying to actually statistically analyse what the risks are and match them up. And people have taken this further and proposed a risk repository of all the risks that are known to be relevant to clinical trials, so you can match them up against everyday life. And they've published a search strategy to do that, and they've published some specimen risks that you might want to be aware of when you talk to ethics committees. So if you want to define minimal risk in a comparison to risks that children would otherwise face, then here's a statement about allergy testing. And these authors propose that we should systematically go through all the risks and do this sort of balance. Other people have taken a different perspective, and this group spent a lot of time analysing Dutch medical research ethics committees and came up with a suggestion that minimal risk of harm applies if empirical data goes to one of these thresholds, so that if the risk of serious harm is less than 1 in a million, then that counts as a minimal risk of harm. So that's a different way of doing it, taking an absolute approach to risk rather than balancing them. Another approach has been put forward by the European Commission with the European Medicines Agency, and this is the document I suggest you consult before going to a European Ethics Committee, particularly in one of the countries that has not put the Oviedo Convention into force. Here's the URL here. And what they do is give practical examples for these risk categories. And so these are available for us in discussion with ethics committees. So this is a top tip. Use this document. There's the URL. You'll find it by Googling Ethical Children at European Medicines Agency. And of course, everything the European Medicines Agency does with children is ethical. So these are the things that the EMA has suggested can be no or minimal risk if appropriately justified. So things like venipuncture and heel prick, which are common in many types of research, would be classified as no or minimal risk, which again is probably more liberal than American definitions. Minor increase over minimal risk would be suprapubic catheterization, an arterial puncture, umbilical catheter, and so on. And these are procedures undertaken during research. And other things which would be greater than minor increase over minimal risk are listed on the right. And so it's possible within Europe, at least, to argue that these can be done specifically for research purposes. You can put that in your protocol. Whether anyone consents to it is a separate issue. So, coming to some case studies, just to weigh up how we apply these criteria. First of all, the single-dose pharmacokinetic study of cough and cold medicines, then the placebo treatment, and then some phase 1 studies. So, in a single-dose PK study of a cough and a cold medicine, should we be doing this to children who don't have coughs and colds? Should we be doing it only to children with coughs and colds? The single dose, which minimizes risks of exposure to the medicine, means that even if you've got the right dose, the child will have no benefit from it. We can design the study in different ways. We can take 2 samples from 50 participants using a sparse sampling approach analyzed with a population PK model. Another way to do it might be to take 10 samples from 20 participants using traditional rich sampling with or without a population PK model. So if you have this person having a cough and a cold, then having blood tests may be part of what they expect when they have a cough and a cold, or it may not be. Or having blood tests may not be part of their everyday routine experience. And then we've got to decide whether exposing 50 participants to a medicine whose dose we don't know, but only giving them 2 blood tests, is minimal as opposed to a different approach. And both of these can be argued, but I suggest that using the EMA's framework is one way to do this. Again, placebo treatments are necessary if we're going to provide adequate research. Children deserve placebos just as much as anybody else, at least until you know whether the medicine works. The placebo group have no potential for benefit from the intervention. Some people argue that children benefit from being in research in other ways apart from the intervention. It's clear that children in randomized controlled trials often receive a better standard of care for other aspects of their care. And children may benefit from education, their parents may benefit from education. These people may benefit from enhanced surveillance of adverse drug reactions, meaning that they come to fewer harm. So that might be a balance. Many people in the States would argue that's not the case, that you should compartmentalize research and that every aspect should be judged on whether it provides benefit or not on the whole package. The other side of it is that you do need to do assessments in placebo-controlled trials to get a fair comparison. So if one group needs a study with ionizing radiation, then the other group should get it as well for a fair comparison. And when there's no benefit to the individuals, then that needs to be justified. Depending on the benefit you may get to the whole group, to the whole population from a medicine that works to treat a particular condition. So here are some examples of ethics committee reasoning taken from a very informative Dutch paper in the Journal of Medical Ethics. The Dutch ethics system has the reputation of being very strict on early phase clinical trials, and if you can get it through a Dutch ethics committee, then you're doing very well from what I've been told. So here's one study that tried to define the maximum tolerated dose, safety, and pharmacokinetics and efficacy of a new cytostatic agent. So this must have been quite a clever study design to do all of these things at the same time. So someone was working really hard and trying to minimize the numbers of patients recruited by doing all of this at the same time. And there are ways to do this if you have the right statistician. They wanted to recruit oncology patients aged 1 to 21 years with a very poor prognosis. Doing this in adults would cause no trouble at all. That when people are at the end of the road for treatment, trying out a new medicine on them is something that some parents, some adults would welcome. The main risks were thought to be over or underdosing of the drug because you don't know the dose, its adverse effects, and anemia, but then that's part of having oncology in many settings. And the burden was 10 visits with 10 venipunctures, 3 one-day admissions, with each with a separate episode for intravenous cannulation and the magnetic resonance imaging procedure. So after lots of discussion, the Ethics Committee definitively rejected this study as being more than minimal risk with no direct benefit to the subjects. Another study related to the definition of safety and the local effect of a potential gene correction therapy in a metabolic disorder. So this is a proof of concept with no intention of benefit for the individuals. So this was looking at children aged between 8 and 16 with a severe and progressive muscle disease. And the main risks were the adverse effects of the drug, gene correction therapy, not just the drug, but the whole process of getting the gene into the body has clearly got unknown risks. And there was a muscle biopsy involved as well. There were 4 visits, 4 venipunctures, magnetic resonance imaging, injections, and the muscle biopsy. These children are old enough to express an opinion, and maybe it's those opinions that we ought to listen to. I suspect the Ethics Committee did that after a long debate and a site visit to ensure that the people doing the study were going to do it well. So this was a non-benefit study which had things which are probably a minor increase over minimal risk that was approved in the Netherlands. And so this is the sort of balance that ethics committees will make. So there were some suggestions that the clinical trials regulation that we've heard about should ban all research that's more than minimal harm, that it should allow minor increase over minimal risk, or it should allow scientific value to be part of the benefit risk assessment. So these are things that our lawmakers are deciding for us. How should we put law into medicine? So when I meet a sick child, how does the law influence my practice? Well, it tells me what to do sometimes. If I have to get consent myself rather than getting consent from other people, I have to do it. But I also have my own standards, and so I do things. I may limit myself more than the law limits me. My professional practice and my ethics codes and other people may limit me, and society may limit me as well. And in the UK, we have a strong governance culture, and that is certainly very limiting, as many people will say. So we may not need to be strict in law, because there are many other ways of being strict and many other safeguards if ethics committees are in place. I guess we have to trust the ethics committees, we have to trust the governance, and it may be that governance sometimes gets it wrong. The benefits and risks of legal definitions are that it leads to standard practice. In the United States, that is not true, and there may not be consensus within the European Union. So, I'm going to wrap up soon. First of all, how should we work with ethics committees? I suggest that we use the EMA guidance about what is minimal risk. 9 times out of 10, that's going to be a very clear justification for what we want to do or to limit our studies designs. So here's that document again, here, And here, this is a fundamental document when I'm writing protocols or joining in with other people. We need to avoid broad statements that sound nice. And the Oviedo Convention had this statement that the benefits should never be included in the assessment of minimal risk. And that is a little bit of an outlier compared to other international documents. And we should work together. We've already heard about the need to harmonise ethics committees, to harmonise the procedures that we all undertake together. And we need to remember it's impossible to develop medicines in one country or even one continent. We need to harmonise across continents, and we need to consider what is ideal and what can be tolerated. And the other bit of this slide that's missing is consultation with families, children and young people, because that has to be central to how we work together, because children and young people have very strong views which are usually correct when it comes to asking them to contribute to research. So we can work together, and the voice that the regulators will listen to is our European network, and there are lots of groups working together to try and help us harmonise our procedures. So I'll stop there. Thank you very much.